Okay, so welcome back to this next video in which we are discussing receptor tyrosine kinases. Okay, so we're currently looking at pathways downstream of receptor tyrosine kinases. Specifically, we're now looking at the PI3 kinase AKT mTOR pathway. Okay, right, so we've seen that what happens is that the receptor tyrosine kinase that is dimerized will have phosphotyrosine residues exposed on its cytoplasmic tail, and these are going to recruit type 1a PI3 kinase enzymes, which will be translocated from the cytoplasm to the underside of the phospholipid bilayer, and once they're there, they will also be activated slightly by the binding to the phosphotyrosine residues. Okay, the regulatory subunit will loosen its hold on the catalytic subunit, and the catalytic subunit will now be able to convert the phosphatidylanocetyl 4,5-bisphosphate molecules into phosphatidylanocetyl 3,4,5 trisphosphate molecules. Okay, now what's going to happen is the phosphatidylanocetyl 3,4,5 trisphosphate molecules are going to recruit and activate a large number of different proteins, but we're specifically going to look at two. Okay, we're going to be looking at the phosphoinositide-dependent kinase 1, PDK1, and also protein kinase B, which is also called AKT. Okay, so we'll start off with phosphoinositide-dependent kinase 1. Okay, so let's say we now have our P3 molecule here. Okay, so let's just draw it out again. And what's going to happen is an enzyme is going to translocate from the cytoplasm to the underside of the phospholipid bilayer by binding to this PIP3 molecule here. Okay, so let's just colour this in. So here are the phosphate groups. Here is the inositol ring in blue here. Here is the glycerol molecule in green. And then here are the long-chain carboxylic acids in orange here. Okay, right. And now what's going to happen is our enzyme, phosphoinositide-dependent kinase 1, is going to bind to the PIP3 molecule. And to do this, it has a special domain, okay? So we've discussed SH2 domains, we've discussed phosphotyrosine binding domains, PTB domains, which are these conserved structures that you find in many different proteins which bind to phosphotyrosine residues. We're now going to see another conserved domain that you find in many different proteins, but this time it's not going to bind to phosphotyrosine residues, but instead is going to bind to PIP3 molecules, okay? And this is known as a pH domain, okay? And pH is short for plextrin homology, okay? So this is a plextrin homology domain. And the reason it's called that is that it's homologous to a domain that was originally found in this protein plextrin, which is found within platelets. Okay, right. This enzyme here, which was previously in the cytoplasm, is called phosphoinositide-dependent kinase 1. And it has this special domain known as a plextrin homology domain, which is then going to bind to the PIP3 molecules once they appear. Okay, so once you get this transient appearance of PIP3 molecules in the inner leaflet of the phospholipid bilayer, it will now be able to bind to these, and therefore it will localize underneath the phospholipid bilayer. Okay, so this is phosphoinositide-dependent kinase 1, okay? And for short, phosphoinositide-dependent kinase 1 is usually abbreviated to P for phosphoinositide, D for dependent, and then K for kinase 1, okay? Right, so the main function of it binding to the PIP3 via its plextrin homology domain is merely to relocate the phosphoinositide-dependent kinase 1 enzyme to the underside of the phospholipid bilayer. Its activity isn't hugely affected by the translocation. Okay, right. Now, it's a kinase enzyme. It's going to phosphorylate serine and threonine residues. So it's a serine-threonine kinase. And it's going to phosphorylate serine and threonine residues in another protein. And this other protein is also going to be recruited to the membrane via a plextrin homology domain, which will bind to PIP3 molecules. Okay, right. So this other enzyme is going to be an AKT enzyme, or a protein kinase B enzyme. So let me draw this here. So once again, we need another PIP3 molecule. So I'll draw this here. So here's the inositol ring. Here are the phosphate groups coming off the alcohol groups of the 3rd, the 4th, and the 5th carbons of the inositol ring. So I'll colour these in purple here. Okay, we'll have the inositol ring once again in blue here. And then we'll have the glycerol molecule in green, and the long-chain carboxylic acids in orange here. Okay, now we are going to recruit a protein kinase B enzyme, or also called AKT. Okay, now there is more than one protein kinase B enzyme, but again, we're not going to fuss over this because effectively they are all going to do pretty much the same thing. Okay, so there are actually 4 different forms of protein kinase B. Okay, so I'll write these down over here. So there's only 3 different genes. The 3 different genes, we'll start off with the 3 different genes, are known as AKT1, AKT2, and AKT3. Okay, now they also have other names. So their other name is protein kinase B, which is usually abbreviated to PKB, and AKT1 is also called protein kinase B alpha, AKT2 is also called protein kinase B beta, and AKT3 is also called protein kinase B gamma. Okay, so these are the 3 genes. Now, in the case of AKT1 and AKT2, both of them only have a single splice variant, whereas AKT3 has 2 splice variants. It has the splice variant which itself is called protein kinase B gamma, and then it has another splice variant which is called protein kinase B gamma 1. And this one's slightly smaller than this one, so let me just remind you of the basic principle of having multiple splice variants. Okay, so let me draw 2 parallel lines here, and these 2 parallel lines are no longer representing the 2 layers of phospholipids within a membrane. Okay, instead these are now going to represent the 2 strands of DNA in a piece of double-stranded DNA. So this is a piece of double-stranded DNA. Now, let's circle this region here, or box this region, and this region is going to be a gene. Okay, so this is a gene. Now, let me just remind you of the central dogma of biology. One of the strands of the DNA will be what's known as the coding strand. Okay, so let's colour this in vivid purple here. Okay, so this is the coding strand, let's say, and then the other strand will be the non-coding strand. Now, the coding strand is the one which RNA polymerase II will work its way along to create a piece of mRNA that is complementary to it. Okay, so RNA polymerase II will work its way along the coding strand to produce a piece of mRNA. Now, this piece of mRNA is not ready to go through the process of translation yet. This is instead known as a piece of pre-mRNA. Okay, so it's not ready to go through translation and actually be turned into a sequence of amino acids yet. Instead, what has to first happen is a process known as splicing. Okay, and the purpose of splicing is to cut certain portions out. So basically, within this piece of pre-mRNA, which is the primary transcript which we have just produced by producing a complementary piece of mRNA to the coding strand, so this is also sometimes called the primary transcript. Basically, within this primary transcript, you are going to have portions which are supposed to be translated and portions which are not supposed to be translated. Okay, so let's say that these 3 portions that I have now boxed, let's say these 3 portions are actually supposed to be translated. The portions that are supposed to be translated are known as the exons. Okay, so these ones which are now in yellow boxes, these are free exons of our pre-mRNA. Okay, and the portions that are in between the free exons, so these 5 portions in between the free exons, 4 portions in between the free exons, these are called the introns. So basically, the reason that this pre-mRNA is not ready to be translated yet is that you don't translate the introns. Firstly, the introns need to be cut out, and then the exons need to be stuck back together, and then you'll translate the later portion of mRNA that just has the exons within it. Okay, so what you do is you cut out the introns, you stick the exons back together, you get a smaller piece of mRNA known as a mature mRNA. Okay, and this process of converting the pre-mRNA into a piece of mature mRNA, this is known as splicing. Okay, right. Now, the concept of different splice variants then comes from the fact that there is, in the case of some pre-mRNAs, different ways of splicing your pre-mRNA, and these will result in different mature mRNAs. Okay, so you can cut up and re-stick together your pre-mRNA in different ways to end up with different mature mRNAs. So there is more than one way in some case to splice the pre-mRNA. Okay, and this means that you can get different pieces of mature mRNA from the same piece of pre-mRNA. So, to give a very nice, easy to understand example of this, let's imagine that this final exon here, exon number 3, is not compulsory. Okay, let's say that it's an optional exon, so you can either include it in your mature mRNA or you don't have to include it. So that means that there are 2 different ways of splicing this pre-mRNA. Either you can have the, you can include all 3 exons here, and let's say that produces this piece of mature mRNA here, or you can include exon 1 and exon 2, but not include exon 3. That will give you a shorter piece of mature mRNA. Okay, these 2 pieces of mature mRNA can now both be translated, and they will give different proteins. Okay, and these are the different splice variants, okay, of a protein. So this is how one gene can produce 2 different proteins. And what happens in the case of protein kinase C gamma is that one of them, it's just like this. It produces one long portion and then one shorter portion, which then doesn't have an end, basically. Okay, so it's identical all the way along this first bit, but then it's just falls short. It just doesn't have this final bit. Okay, and that's exactly what happens in the case of protein kinase C gamma 1. Okay, protein kinase C gamma 1, this splice variant here is just a shorter version of protein kinase C gamma. Sorry, protein kinase B gamma. Okay, so protein kinase B gamma 1 is a shorter version of protein kinase B gamma. Okay, right. So, there are these 4 different protein kinase B enzymes then. There is protein kinase B alpha, there is protein kinase B beta, and then there are the 2 splice variants of protein kinase B gamma. Okay, protein kinase B gamma itself, and then the short version, protein kinase B gamma 1. Okay, now let me just discuss with you the structure of these protein kinase B enzymes. So basically, they are a single polypeptide, so here's the amino group. Then they're going to have this important domain which is going to bind to the PIP-free molecules. Okay, and this is going to be the plextrin homology domain, and I should colour it in the same colour as I coloured it in on phosphoinositide dependent kinase. So here in turquoise, this is our plextrin homology domain. So this is our pH domain. Okay, then after the plextrin homology domain, you have the catalytic domain of protein kinase B here. Okay, and this is the portion which will be a serine-threonine kinase. Then after the catalytic domain, you have what's known as a regulatory domain here. And then you end with your carboxylic acid tail here. Okay, right. So, let me colour in these different portions. So here's the catalytic portion, which is actually going to catalyse the addition of phosphate groups onto serine and threonine residues within proteins. So let me just label that up as well. This is the catalytic domain. okay, of the protein kinase B protein. And then finally, you have a regulatory domain. Now, all four forms of protein kinase B all have the same structure. Protein kinase B gamma 1 will have a shorter regulatory regulatory domain than protein kinase B gamma. Okay, so in orange here, this is the regulatory domain. Okay. Right, so how do you then activate protein kinase B enzymes? Well, they are going to translocate from the cytoplasm up to the underside of the phospholipid bilayer via the binding of their plextrin homology domain to the PIP-free molecules. Okay, but that's not going to result in their activation. In order to activate protein kinase B enzymes, you have to phosphorylate a threonine residue which is within the catalytic domain. So T is the single letter amino acid code for threonine. Okay, so you've got a threonine residue within the catalytic domain of your protein kinase B enzyme, which needs to be phosphorylated in order to activate the protein kinase B. And in most protein kinase Bs, you also have a serine in the regulatory domain, which also needs to be phosphorylated. So you need both of these to be phosphorylated. So S is the single letter amino acid code for serine. Okay, now, here is one significant thing about protein kinase B gamma 1. It's the only one that doesn't have this serine in the regulatory domain that needs to be phosphorylated in order to activate it. Because remember, I told you it has a shorter regulatory domain than protein kinase B gamma, and its regulatory domain is actually so short that it doesn't contain this serine which requires phosphorylation. Okay, but all of the other three, protein kinase B alpha, protein kinase B beta, and protein kinase B gamma, they all have both a threonine in their catalytic domain and a serine in their regulatory domain, and in order to be activated, they need phosphorylation of both the threonine and the serine. Protein kinase B gamma 1 only has the threonine in the catalytic domain. Okay, right. So, what's now going to happen is that once you've translocated the protein kinase B up to the underside of the phospholipid bilayer, it's now going to be in the same place as the phosphoinositide-dependent kinase 1 enzyme. Okay, now, another important thing to say is that when protein kinase B is recruited up to the underside of the phospholipid bilayer, this binding does change conformation a little bit, okay? And it changes the conformation such that this threonine here in the catalytic domain becomes an excellent substrate for phosphoinositide-dependent kinase 1, okay? So, before this conformational change, this threonine wasn't a very good phosphorylation site. It wasn't a very good target for PDK1. But now, after the conformational change, it's a fantastic phosphorylation site for this PDK1 enzyme. So what's going to happen is that PDK1 is going to phosphorylate that threonine residue. Okay, so note this is kind of like role reversal here. Okay, usually the sort of archetypal pathway would be that this enzyme comes up here, binds onto this becomes activated, it then phosphorylates the threonine. In this case, it's actually that the substrate for the phosphorylation, which is the protein kinase B enzyme, is going to bind to PIP3, change its conformation to become a better substrate, and now it's going to be phosphorylated by a kinase enzyme that wasn't really activated that much by binding to the PIP3. So it's the protein kinase B that changes conformation to allow phosphorylation by phosphoinositide-dependent kinase 1. So, PDK1 phosphorylates this threonine on the catalytic domain. Now, for protein kinase B gamma 1, that's enough, but for the other three, it's not. You also need the phosphorylation of the serine before they will become active. Now, this is still a very controversial area. Okay, it is not well understood at all how that serine becomes phosphorylated and what the significance of its phosphorylation is as far as pathways go. Okay, but it is an essential thing that serine needs to be phosphorylated by something, and it doesn't seem to be being phosphorylated by phosphoinositide-dependent kinase 1. Okay, so something else phosphorylates that serine. Okay, and when both the threonine in the catalytic domain and the serine in the regulatory domain are phosphorylated, the protein kinase B enzyme is then going to detach from the PIP3 molecule. Okay, so it will detach from the PIP3 molecule, it will then go into the cytoplasm, and it's then going to target the mTOR complex 1. Okay, which we'll come on to now. Okay, so what's now happened is our protein kinase B enzyme has detached from the membrane now that it's got both of these phosphate groups, both of these phosphorylation sites phosphorylated. Okay, so let's draw our now active protein kinase B enzyme. So it's now got the threonine within the catalytic domain phosphorylated, and it's also got that serine in the regulatory domain phosphorylated. And of course, I'm talking about the more general protein kinase B, which does have this serine in the regulatory domain, rather than just the protein kinase B gamma 1. Okay, so here in orange, this is our regulatory domain. Okay, and we've got this phosphate group coming off the serine here. Okay, and also the threonine now as well. Then we've got our catalytic domain, which I'll colour in turquoise here. Okay, and this is now going to be active. This is going to be phosphorylating serine and threonine residues within proteins. And then we've also got our plexgen homology domain that is now no longer bound to the PIP3. Okay, so it's in the cytoplasm. And it's now going to phosphorylate serine and threonine residues on downstream targets. So it is now going to activate something known as the mTORC1, okay, which we will come on to in the next video.